Hello guys, I am Shubham from Tutorials. This video is about translation PYQs. Okay, so before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but it will help us grow. This will be the part 1 of this translation PYQs series of CSIR net, right? So first question, different arms in tRNA structure are shown in column A. The specific signatures associated with the different arms are shown in column B. Okay, so match the following है. This is very easy to solve, right? See, जो C है ना, T5C arm, this consists of pseudouridine. Okay, so C4. C4 is present in option 2 and option 4. Okay, then D arm, dihydrouridine. Okay, D1. So option 4 answer होगा. Okay, rest is acceptor arm, 8 base pair stem and CCA sequence. And anticodon arm is 5 base pair stem, A2 and B3. Got it? This is a tRNA structure, okay? Clover leaf model, right? And this is a 3D structure. You can see this is acceptor stem with CCA sequence. Then यहाँ पे anticodon loop, variable loop. Then this is pseudouridine loop. D loop is dihydrouridine loop. Right? Same here you can see this in the three-dimensional structure. तो जो ये क्लोवर लीफ मॉडल और क्लोवर लीफ स्ट्रक्चर जो है, दिस इज अ सेकेंडरी स्ट्रक्चर. दिस इज अ सेकेंडरी स्ट्रक्चर. Okay. Next question. Following observations were made about tRNA. Right? tRNA encoding genes cluster at specific regions of the human genome. Okay, suppose we don't know this, we will keep this on hold. Okay, A statement हमें आइडिया नहीं है, होल्ड पर रखेंगे. B, 5' end of mature tRNA is generated by RNase-P mediated cleavage. ये करेक्ट है, राइट? जो एंड है 5' end, that can be produced by RNase-P mediated cleavage. Okay, हो सकता है. So this is correct. Option C, modified bases may confer increased stability to tRNA. May confer increased stability. Yes. जो bases है modified like pseudouridine, dihydrouridine, ये एक modified bases है. So this can increase the stability of tRNA. So C तो हमें correct चाहिए ही, right? And B also. So option 2 and option 4 will get eliminated. Okay, now see CE and CE common है. What is E? Suppressor tRNAs compete with the release factors to read the termination codon. Yes, suppressor मतलब क्या? Suppress करेंगे translation process को, right? So they will compete with release factors. Okay, release factors जो release करते हैं, right? After translation is completed and recycle the process. So this is absolutely correct. CE confirm correct होगा. Now from B and A, जो A है tRNA encoding gene cluster at specific regions of the human genome. अगर B correct है, already we have marked B is correct, so option 3 correct होगा, because B जो है 5' end generation can be result of RNase-P mediated cleavage, so ये correct होगा. And जो A है that will be wrong, right? Means what? जो tRNA encoding genes है स्पेसिफिक रीजन पर क्लस्टर नहीं होते, राइट, मतलब एक ही रीजन पर नहीं प्रेजेंट होंगे, दे विल बी प्रेजेंट इन मल्टीपल रीजन्स, राइट. And if we see the option D, modified bases of tRNA do not affect the pairing of oval pairing. Okay, तो जो bases modified होते हैं उससे pairing affect नहीं होगा, ऐसा बोल रहे हैं स्टेटमेंट, which is obviously will be wrong, right, because oval pairing affect होगी अगर bases modified है तो. Okay, so you can solve like this also, that D will be compulsory wrong, so 2 and 4 will be eliminated and 1 and 3 में से आंसर होगा. C E तो कंफर्म करेक्ट है, देन A and B में जो B है that is 100% correct. Okay, because 5' end generation by RNA speed mediated cleavage is right, because RNA is a nucleus cut ही करता है, right. Then the next question, following statements are made with reference to the fact that tRNAs are known to possess T in their sequence. T sequence में present है, okay, in the tRNA structure T is present, otherwise RNA में T present नहीं होता, U is present, right, uracil. So how T came? Let's see the possible reasons. RNA polymerase 3 utilizes TTP as one of the substrates. No, TTP को यूज नहीं करता RNA polymerase 3, right? ये तो DNA यूज करता है, DNA polymerase for DNA synthesis. Like any other RNA polymerase 3 also utilizes UTP. But when the RNA pol 3 reaches a loop structure, it binds to S-adenosine methionine to methylate C5 position of the incorporated U to result in thymine in a co-transcription manner. Means what they are saying कि पहले U ऐड होगा, उसके बाद इसका मॉडिफिकेशन होगा by SAM, right, S-adenosine methionine, जो मिथाइलेट करेगा U को, right, and this will get converted to T then. Okay, so let's see with the other options. I am confused in this because ये possible है, मतलब हो सकता है, this can be occurred, so we will keep this on hold, right, hold पर रखेंगे statement को. Let's check with other statements. During transcription of tRNA genes at the designated position, DNA polymerase replaces RNA polymerase 3 to incorporate T in the tRNA transcript. ये कुछ भी है statement. Okay, so this will not be the answer because DNA polymerase RNA polymerase को replace नहीं कर सकता. DNA polymerase के requirements अलग है, RNA polymerase के requirements अलग है, right. DNA polymerase is for replication, RNA polymerase is for transcription, right, so the requirements are different. Okay, so A and C will be wrong. A option 1 में है, C option 2 में है, 3 में है, right. So B can be answer, but B के साथ C नहीं है, right, answer. So ये eliminate हो जाएगा option 2 and वैसे ही फोर्थ ऑप्शन करेक्ट होगा, राइट, बिकॉज सी कंफर्म रॉन्ग होगा. आरएनए पॉलीमरेज को डीएनए पॉलीमर रिप्लेस नहीं कर सकता इन एनी केस. ओके, so let's check the other options. A specific methyl transferase utilizes a methyl group, right, methyl group यूज करेगा donor to post transcriptionally modify the specific U residues into T residue. Yes, so you can see ये statement similar है to the statement B, राइट, मीन्स मिथाइल ग्रुप दोनों को यूज करके मिथाइल को ट्रांसफर होगा मिथाइल का, एंड देन देयर विल बी मॉडिफिकेशन ऑफ यूरासिल टू थायमिन. सो दिस विल बी करेक्ट, राइट, डी इज करेक्ट. यूरासिल टू थायमिन कन्वर्जन occurs in large number of tRNAs in the T5C loop. So this you have to remember. यूरासिल टू थायमिन कन्वर्जन है, कहाँ पर होता है, this loop, T5C loop. Okay, so like this you can solve the questions by elimination, right. एक स्टेटमेंट या दो स्टेटमेंट ऐसे होते हैं that you can confirm, say that these are the wrong ones. Okay, wrong statements आप choose कर सकते हो and then use them to select the correct answer. Okay, so here we have to find the correct statements only. This is also very important. आपको ध्यान देना है exactly क्या आपको आइडेंटिफाई करना है इस क्वेश्चन में. कभी कभी करेक्ट करना होता है, कभी कभी इनकरेक्ट करना होता है. So you have to pay the attention carefully. Amino acyl tRNAs are escorted to the ribosome by the elongation factor. राइट, अभी ये elongation से है क्वेश्चन ट्रांसलेशन, तो इसका डायरेक्ट आंसर है AFTU ऑप्शन 3. ओके, सो हमने पूरा ट्रांसलेशन प्रोसेस डिटेल में डिस्कस किया है इन मॉलिक्यूलर बायोलॉजी. राइट, मॉलिक्यूलर बायोलॉजी हमने पूरा डिस्कस किया है विथ टिप्स एंड ट्रिक्स टू रिमेम्बर. राइट, बिकॉज वो कॉम्प्लेक्स होता है. आई हैव ट्राइड टू मेक देम इजी. ओके, so please watch that video and then solve these questions. Each amino acyl tRNA synthetase is precisely able to match an amino acid with the tRNA containing the correct corresponding anticodon. Okay, most organisms have 20 different tRNA synthetase. However, some bacteria lack the synthetase for charging the tRNA for glutamine. Okay, with its cognate amino acid. Cognate means it's appropriate. So GLN is glutamine, GLU is glutamate. Okay. How do these bacteria manage to incorporate glutamine in their proteins? मतलब glutamine का tRNA नहीं है, tRNA synthetase. So how glutamine is incorporated? Choose the correct answer. Glutamine is not present in newly synthesized bacterial protein. Post-translational modification converts glutamate to glutamine at the required sites. Not present in the newly synthesized bacterial protein. This is completely wrong statement. In this bacteria, the amino acid tRNA synthetase specific for tRNA glutamate also charges glutamine, tRNA glutamine with glutamine. Yes, ये correct है, partial correct है, partial. Let's see the other statements also. In this bacteria, the amino acid tRNA synthetase specific for tRNA glutamate also charges tRNA glutamine with glutamine. A second enzyme then converts the glutamate of the charged glutamine to glutamine. Yes, ये मोस्ट एक्यूरेट है. फिर 2 and 3 में जो फर्स्ट सेंटेंस है ना सेम दिया है, बट जो नेक्स्ट सेंटेंस है वो थर्ड स्टेटमेंट में प्रेजेंट है. सो ये आंसर होगा. ओके, सो देयर विल बी कंफ्यूजन बिटवीन 2 and 3, राइट, बिकॉज बोथ आर करेक्ट, बट 3 इज मोस्ट एक्यूरेट बिकॉज ग्लूटामेट वाला tRNA सिंथेटेज ही ग्लूटामाइन को चार्ज करता है. राइट, जस्ट रिमेंबर दिस इज रिपीटेड क्वेश्चन. राइट, एंड ये इंपॉर्टेंट है बहुत. सो दिस आर सेम. जो ग्लूटामाइट वाला tRNA है वो ग्लूटामाइन को चार्ज करेगा. ओके, बट व्हाट हैपेंस, दैट इज अ सेकेंड एंजाइम दैट कन्वर्ट्स, मतलब ग्लूटामेट का कन्वर्जन करेगा अनदर एंजाइम. राइट, so glutamate to glutamine gets conversion by a enzyme. Next statement, in this bacteria the amino acid tRNA synthetase charges tRNA glutamate with either glutamate or glutamine, according to the requirement during protein synthesis. No, ऐसा नहीं है. राइट, मोस्ट एक्यूरेट स्टेटमेंट इज 3. तो ये भी पार्शियल करेक्ट बोल सकते हैं हम लोग. वाई, बिकॉज tRNA ग्लूटामेट वाला ही ग्लूटामेट एंड ग्लूटामाइन को चार्ज करेगा. बट जो प्रॉपर प्रोसेस है दैट इज डिस्क्राइब इन द ऑप्शन 3. नेक्स्ट वन, बैक्टीरियल राइबोसोम कंसिस्ट ऑफ 30S and 50S ribosomal subunits. Right, the translating monosome has a sedimentation value of. Okay, so bacterial ribosome का केस है 70S. राइट, large subunit 50s, small subunit 30s. ओके, इसका टोटल होता है 80s, बट एज ए यूनिट हम 70s बोलते हैं, राइट? वाई सो? ऑप्शन 1 एंड ऑप्शन 2 में से ही आंसर होगा। 3 में तो 80s है विच इज यूकैरियोटिक केस, तो ये नहीं होगा, 50s भी नहीं होगा। ओके, इजी। अभी देखेंगे विच वन इज द एग्जैक्टली करेक्ट ऑप्शन। 70s because a fixed set of ribosomal protein totaling to a value of 10s are removed when 30s and 50s subunits interact with each other. S stands for Swedberg, okay, in the sedimentation, right? Swedberg unit. So 10s का ribosomal proteins are removed as per the first statement. तो ऐसा नहीं होता, राइट? प्रोटीन का रिमूवल, नो, so this will be wrong. तो ऑब्वियसली ऑप्शन 2 है करेक्ट होगा, because the interaction between the two subunits 30s and 50s excludes some surface area, decreasing the overall resistance of movement through the medium, right? मतलब सरफेस एरिया को एक्सक्लूड करते हैं, अवॉइड करते हैं, एंड उसकी वजह से जो रेजिस्टेंस है ओवरऑल डिक्रीज होता है, राइट? सो दिस इज अ प्रॉपर statement, reason why 70s is the bacterial ribosome consisting of 30s and 50s subunits. Alright, so there is a case of surface area. You should remember this. Alright. Why PYQ solving is important? Because question repeat होते हैं, right? In the same exam as well as other exams like DBT, ICMR, GATE. Okay, questions get repeated. And you will see them in the channel as well because I have solved so much of PYQs. Let's see others option also. 80s because the monosome consists of one subunit of 30s and one subunit of 50s. No, because 80s तो eukaryotic case है, right? Consisting of 60s and 40s. Last subunit, small subunit. So this is of eukaryotes. 50s तो subunit last subunit का है because the sedimentation of the combined monosome is determined by the sedimentation of the last subunit. Okay, no. So हमने आंसर ऑलरेडी किया है. Let's see the other. Precise recognition of tRNA by the cognate amino acid tRNA synthesis is crucial for the fidelity of protein synthesis, right? In the context of amino acylation of tRNA alanine with its cognate amino acid tRNA synthesis and based on studies on the molecules of Escherichia coli origin, following statements are made. Which one of the following statement is incorrect? Okay, see now, ये जो अमाइनो एसिड के रिगार्डिंग क्वेश्चंस है, ये हमने प्रॉपरली वन बाय वन ऐसे डिस्कस नहीं किया है because there are lot of variations seen. Okay, so this we are seeing from the questions, right? How the questions are asked and the points to remember. Okay, so incorrect statement we have to identify. So this is the case of alanine. You have to remember, right? Anticodon of tRNA alanine makes important contribution to the specificity of its amino acylation by alanine tRNA synthesis. तो ये ही incorrect statement है because anticodon का रोल यहाँ पे important नहीं है, right? Why? I will tell you. Because the rest three answers are correct. Mutational analysis have shown that for amino acylation of the tRNA alanine by alanine tRNA synthetase, presence of a oval pair in the acceptor stem G3U70 is the most crucial element. Okay, so you have to remember this statement, very important. This is very important. ये जो है ना acceptor stem में इसका होना, this is the most crucial element, right? At the acceptor stem G3U70. Okay, then amino acylation of tRNA alanine by alanine tRNA synthetase occurs even if anticodon of tRNA alanine is mutated. So ये statement correct है, and that is why जो option 1 है incorrect होता है. Got it? Because anticodon में अगर mutation है तो भी alanine attach होता है. Amino acylation with the alanine takes place. Then the fourth statement is a microhelix lacking a clover leaf structure. See, अभी tRNA structure में clover leaf structure में problem है. Microhelix is lacking a clover leaf structure and harboring only the acceptor stem sequence of the tRNA alanine is specifically amino acylated by alanine RNA synthetase. Okay, so ये भी correct है. Means what it is given, you have to understand the statements and remember this. Okay, because ये question repeat हो सकता है, and because this has repeated 2022 में. तो microhelix मतलब clover leaf structure present नहीं है. ओके, एंड एक्सेप्टर स्टेम जो है, राइट, 3 प्राइम में जो एक्टिव एक्सेप्टर स्टेम होता है CCA. ओके, तो ये जो है ना एक्सेप्टर स्टेम वाला, इसको अगर हार्बर किया तो अमाइनो एसिलेशन होता है बाय द alanine, राइट. सो एक्सेप्टर स्टेम इज इंपॉर्टेंट. व्हाट यू कैन अंडरस्टैंड, एक्सेप्टर स्टेम इंपॉर्टेंट है बट एंटीकोडॉन इंपॉर्टेंट नहीं है, राइट. मतलब अगर एंटीकोडॉन में म्यूटेशन है तो भी अमाइनो एसिलेशन होता है. So this is a special case of alanine. You have to remember. Okay. Next one, presence of selenocysteine in protein in E. coli is a consequence of. अभी selenocysteine coded by UGA codon, and which is a stop codon also, right? अभी what is the case here? Post translational modification of cysteine present in special structural region of the proteins by cell B and cell C, no. Post translational modification of serine present in special structural region of the protein by cell B and cell C, no. Amino acylation of a special tRNA selenocysteine by serine tRNA synthetase with serine. Okay, first serine add होता है, followed by further modification of attached serine to selenocysteine, right. serine का modification होके selenocysteine होता है, देन उसका ट्रांसपोर्ट होगा टू द राइबोसोम बाय सेल बी. So this is correct statement. Okay, this is also you have to remember, right. There is a special tRNA selenocysteine where serine gets added first, right. Serine followed by further modification of the attached serine to selenocysteine, and जो इसका ट्रांसपोर्ट होगा to राइबोसोम बाय सेल बी. ओके, एंड जो नेक्स्ट ऑप्शन है अमाइनो एसिड ऑफ स्पेशल tRNA selenocysteine by serine tRNA synthetase with selenocysteine, नो. डायरेक्ट selenocysteine से नहीं, फर्स्ट serine देन उसका मॉडिफिकेशन, देन उसका ट्रांसपोर्ट. ओके, सो ऑप्शन 3 आंसर है. नेक्स्ट वन, many organisms encode only 18 amino acyl tRNA synthetase. These organisms lack amino acyl tRNA synthetase that uses asparagine or glutamine as one of the substrates. Okay, repeated question. For direct amino acylation of tRNA, asparagine and glutamine respectively. Okay, मतलब tRNA present नहीं है. Not tRNA, actually tRNA synthetase. Amino acyl tRNA synthetase which do the amino acylation, right? Amino acid को add करना on the tRNA. So asparagine, see you have to note this, asparagine and glutamine दोनों अमीनो एसिड्स के लिए tRNA synthetase नहीं होता in the bacteria. So we have answered this question previously. Same answer है. Which one of the following statements represent the correct option? So basically this is you have to note negatively charged जो अमीनो एसिड्स है, ग्लूटामेट एंड एस्पर्टेट, उसका केस है ये, right? Like this you can remember. The organisms lacking asparagine RNA synthetase and glutamine RNA synthetase lack asparagine and glutamine in their proteins. No, confirm wrong है. बोल रहा है कि प्रोटीन्स होते ही नहीं है, मतलब ये अमीनो एसिड्स ही नहीं होंगे प्रोटीन्स में. So this is confirm wrong. In these organisms, selected aspartame glutamate residue in the proteins are post-translationally modified by a regulated mechanism. Somewhat correct. Okay, this is partial correct, but let's keep on hold. In these organisms, the tRNA asparagine and tRNA glutamine first amino acylated by asparagine, aspartate and glutamate. ASP is aspartate and glutamate. So first ये add होगा, and then aspartate and glutamate attach to the tRNAs are modified to asparagine and glutamine. Then पहला इसका attachment and then modification to asparagine and glutamine respectively. So this is confirm correct. The next statement है, in these organisms, the precursors of mRNA that encode aspartate RNA synthetase and glutamate RNA synthetase are alternatively spliced to generate asparagine RNA synthetase and glutamine RNA. No, alternative splicing का कुछ भी रोल नहीं है इसमें. Next question. So basically this question is of now genetic codes है ना, codons and the amino acid that you have to remember for these questions. Okay, I will show you how you can solve very easily. What would be the tripeptide produced by translation of the transcript produced by the following DNA sequence? अभी यहाँ पे DNA sequence 3' to 5' दिया है. So we know that जो translation होता है that is 5' to 3'. तो इसका हमें conversion करना पड़ेगा. Okay, so 5' will be T T C, right? Then A T G A G T. Okay, got it? अभी just replace this by U. because in the RNA T is replaced by U. Got it? Now this is more simple to solve. See here, ये codon होगा first, ये second and ये third. Right? So ये second amino acid. Basically this is a start codon AUG which codes for methionine. So middle में methionine present only in option 4. See. Okay, this is how you can solve quickly. Next question again. This is little bit complex, you can say. Imagine the following RNA sequence is translated in the mammalian cytosol and mitochondria. Okay, cytosol का भी देखना है, mitochondria का भी देखना है. RNA sequence is given. Following are some putative peptide sequences. अभी यहाँ पे most simple to solve is AUG again methionine होगा. Methionine present everywhere. Okay, AUA suppose we don't know. CUG भी नहीं पता. So this is second position, third position. UGA is a stop codon. Right? UGA stop codon होगा. तो यहाँ पे कुछ कोड अमीनो एसिड नहीं होगा. So I think A will be the correct option for cytosol case. Right? And about mitochondria हमें देखना पड़ेगा. Okay, so let's see now in the table. AUG methionine confirm है. Now what is AUA? First position A, U, AUA. Okay. Isoleucine. Right, see this. So isoleucine होगा. Isoleucine. Then leucine, ये correct होगा. CUG is leucine. CUG is leucine. Correct? And UGA is a UAG is a stop codon. No, it's UGA. Stop codon. So in this case it is there. But in the mitochondria case, this codes for tryptophan. See. Right? Mitochondria. You can make a screenshot of both of these charts. Right? So this is absolutely wrong, as I told you already. यहाँ पे tryptophan होगा. See, there is one confusion here. यहाँ पे isoleucine है and यहाँ पे methionine है. Again. So let's see. AUA in the case of mitochondria is what exactly? The AUA in the case of mitochondria methionine है. Special case है. Highlight किया है. So you can remember this. Right? UGA codes for tryptophan and AUA codes for methionine. So if you know that, you can answer this question easily. So option 1 again नहीं होगा, ये होगा. So A2, option 2 is the correct one. A2, find out the correct combination peptides made in the cytosol and mitochondria. Okay, so this is how you can solve. I will erase this so that you can make a screenshot. Both of these tables are very important about the genetic code. This is the main cytosol nucleus case and mitochondria wala this is little complicated to solve. Okay, but let's see how. Given below is a partial coding sequence of a gene 5' to 3', so no conversion needed. Which one of the following polypeptide cannot, see this is very important, cannot be produced by transcription and translation of the above DNA sequence in any of the 3 possible reading frames. Okay, दिया है 3 reading frames हमें देखना है, right? So first reading frame will be starting from AAT, GGA, CGC, ATG, okay, then TGT, TGA, TGG. Got it? This is the first reading frame. तो यहाँ पे ATG, AUG है, which is methionine again. तो methionine ये first option से confirm होगा. Okay. Based on the options, what I can see is जो one option है ना, 1 and 2, इसमें ये सिलेक्शन होगा. तो फर्स्ट वन में मिथियोनिन है 4th पोजीशन पे, एंड जो सेकंड है उसमें फिनाइल एलानाइन सिस्टीन है. तो फर्स्ट इज व्हाट, यू हैव टू रिप्लेस द टी विथ यू. ओके, सो ए यू का क्या होगा देखेंगे हम. ए यू ना, so this is A, this one in the second position, C asparagine. Okay, so this is confirm again. So this यहाँ पे asparagine होगा. Second जो है GGA. This is time taking questions, right? And remembering this table is little bit. I am not that good in this. So this is G, this is G, and then A will come here. This is glycine. So glycine. So you can see here, यहाँ पे glycine है, ये arginine होगा. CGC, CGC, arginine, right? Arginine. So this is confirmed reading frame. Okay, confirm हो रहा है. अभी जो सेकेंड रीडिंग फ्रेम होगा, स्टार्टिंग का ए कैंसिल करो, देन गो विथ एटीजी. एटीजी मिथियोनिन, एटीजी मिथियोनिन, दिस इज मिथियोनिन. सेकेंड इज GAC, GAC. 2 अमीनो एसिड से देखेंगे हम, राइट? यू कैन चेक फॉर रेस्ट वन. दिस G, दिस A, एंड देन C. यू कैन सी यहाँ पे एस्पराजीन है. तो दिस इज एस्पराजीन. तो यस, ये भी कंफर्म है. अभी 2 अमीनो एसिड को कट करो स्टार्टिंग में, एंड थर्ड रीडिंग फ्रेम विल बी TGG, means UGG, and ACG. Let's see UGG क्या है. UGG is tryptophan. Okay, this is tryptophan. And ACG, यहाँ पे A है, यहाँ पे C है. ACG is threonine. तो यार it is threonine. तो यस, ये भी रीडिंग फ्रेम है. ऑप्शन 2 ही इनकरेक्ट है. So based on my logic, as I said while starting this question, confirm second year incorrect है, because that is the 1 and 2 ऑप्शन सेम है. स्टार्टिंग का जो अमीनो एसिड है एस्पराजीन, I think I have confused with asparagine and aspartate. Just remember ASN asparagine होता है और ASP aspartate होता है. GLU ग्लूटामेट होता है, GLN ग्लूटामाइन होता है, right? So how I solve summary? So ATG है in the first reading frame, that will be methionine, ओके, एंड ये सिर्फ वन ऑप्शन में है एंड जो सेकंड ऑप्शन में प्रेजेंट नहीं है, तो ऑब्वियसली ऑप्शन 2 इनकरेक्ट होगा. एंड आफ्टर चेकिंग विथ अदर रीडिंग frames, that is getting confirmed. Okay, so this is how you can solve questions quickly and easily, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel, and stay tuned for the upcoming videos. Thank you.